Hey, what's up? And welcome back to another edition of Purple Bear Biology. In this conversation, we're going to discuss some cellular biology. We will focus our conversation on the important parts of the cell that we need to understand for some physiology concepts that we'll cover later in other conversations. And we need to discuss structure and functions inside of the cell so that we understand how they replicate and where genetic diversity comes from. We also need to understand what happens when the cell cycle breaks and is not regulated anymore. Let's start by making sure we understand what a cell is. Check this out in the slides.
Cells are the basic building block of life. One or more cell types combine together to make tissues, and tissues combine together to make organs, and organs combine together to make organ systems. While there are diverse types of cells, and each of them serve a particular function throughout the body, most share these four common abilities. Cells must have the ability to acquire energy and use it to survive. Cells must be able to actively transport nutrients into the cell and waste out of the cell. Cells must be able to communicate with other cells around them to make sure that tissues, body systems, and even multiple body systems can work together to maintain our survival. And then lastly, cells need to be able to produce new cells to replace old or dead ones. Okay. So now that we know what a cell is, let's talk about the important parts of a cell that you will need to understand for our future conversations. Here is a list of the different parts that we are going to discuss. The plasma membrane, that includes a separation from what is intracellular fluid and extracellular fluid, mitochondria, nucleus, and DNA, and then the Golgi apparatus and the endoplasmic reticulum. Starting on the outside, and working our way into the cell, the first structure we would encounter is the plasma membrane. This membrane is a composition of lipids, proteins, and sugars responsible for establishing and regulating the inside and the outside of the cell. Fluid found on the outside of the cell is referred to as extracellular fluid, while fluid found on the inside of the cell is referred to as intracellular fluid, or sometimes called the cytoplasm. While the plasma membrane separates the two environments, it is important to realize that it is not rigid. Rather, it moves like a fluid and is what we call selectively permeable. Selective permeability means that the membrane only allows specific chemicals or substances to enter and leave. Why do we care about any of these things? Well, the membrane helps regulate the inside of every cell and maintains the internal environment even when the external environment changes. Maintaining a stable internal environment will be vital for a cell to survive and function correctly. Now let's discuss the specialized structure called mitochondria. Many of us learned about mitochondria in grade school as the powerhouses of the cell. Indeed, its primary function is to help facilitate the creation of usable energy for the cell. You can think of it a lot like money. If you have US dollars and are currently in a rural area in China, you're going to find your money is not immediately useful. The money must first be converted to Chinese currency to be used to purchase things. Similarly, the energy that you get from consuming foods is not immediately accessible to your cells. Instead, it must be converted to a usable form, and mitochondria are one of the primary steps to converting to a cellular energy currency called ATP. We will discuss more about ATP and how is it used when we talk about the chemistry of anatomy in our next discussion. As we dive deeper into the cell, we get to the nucleus that wraps around our genetic material and protects it. This genetic material, called DNA, serves as the directions to create copies of the cells and the structures needed to maintain them. While we are talking about DNA, we should discuss the fact that in order for a cell to divide and make a copy of itself, the DNA must get copied too. Additionally, this DNA serves as a set of instructions to make proteins. We will talk all about proteins in our chemistry discussion, but these molecules are a lot like the workers of the cell. They can help combine molecules, break down compounds, and regulate transport across the plasma membrane. Without functioning proteins, a cell cannot survive. Proteins are built through a two-step process called transcription and translation. Transcription takes place in the nucleus and occurs when the cell converts DNA into a usable message called RNA. Overall, RNA is very similar to DNA with a few subtle changes. But most importantly, RNA can be transported out of the nucleus. Once outside of the nucleus, the RNA message is used as instructions to build a long chain of chemicals. This chain will be the proteins, but it is not functional until it has been folded into the correct shape. This is kind of like how metal can be useful to make things like spoons for eating. The metal in its raw form is not all that useful for eating, but when molded into the shape of a spoon, gains functionality and usefulness. Shaping of proteins brings us to the last two structures we are going to discuss, the endoplasmic reticulum and the Golgi apparatus. These structures serve as the shapers and packagers of proteins. They help fold the proteins into functional forms and package the proteins for transport around the cell. Okay, so now that we have an understanding of some of the general parts of a cell, let's focus in on our last topic, cell replication and cycles. Cellular replication is a complicated topic, and how exactly DNA gets copied involves multiple steps and lots of different proteins. Instead of getting bogged down in all that is happening in every single stage and the machinery that is needed to complete the tasks, I want to stick to two main points. First, in order for a cell to divide, it must make a copy of its DNA. When information is copied, there are chances for mistakes to take place. Mistakes in DNA are often called mutations, and if not repaired, can have catastrophic effects for the newly forming cell. Most of the time, these mutations are either harmful or have no effect at all. But rarely, the mutations result in new beneficial characteristics in the newly forming cell. If the mutation occurs while making gametes, like sperm and eggs, then the new characteristic can even be passed on to the next generation and be present in the offspring. Our second main point is about cell cycles. Cell cycles have multiple stages that take place to accomplish replication and cellular division. Throughout these stages, there are checkpoints to ensure no mistakes or errors may have occurred during DNA replication. If an error is discovered in the DNA or in how the DNA is getting separated between the two newly forming cells, then the cell will trigger a process called apoptosis, where the cell kills itself, kind of like a self-destruction protocol. Why in the world would it do such a thing? Well, if cells develop mutations in the replication cycle, it can lead to uncontrolled replication, and this results in cancerous cells that can develop into tumors. One crazy, cool, and important thing to remember when we talk about cancer cells is that they often retain the properties of the original cells. So, if a cell like this one normally produces hormones that are distributed around the entire body, if it becomes cancerous, then all of the cells that are created through the uncontrolled cellular replication potentially can produce that same product. This means that the hormone product may go from being what is well regulated to out of control or having an overabundance of this particular chemical. This is one of the primary ways that cancers can have full body, also known as systemic effects.
Wow, that is all pretty awesome, but it's definitely a lot of information to take in all at once. So let's summarize the major points that we talked about. First, cells make up the building blocks of life and have specific functions based on the cell type and location that they're found. Second, cells are made up of numerous structures to perform their daily functions. Third, cell replication involves genetic duplication and mutations can sometimes occur. Uncontrolled cell replication can lead to cancer and systemic problems due to overexpression of particular traits. I hope all of that makes sense. If you have any questions or comments, be sure to drop them in the comment section below. If you like the video, make sure that you hit the like button and to subscribe so that you can continue to follow the channel for more cool videos on anatomy. Thanks for stopping by and see you next time.